loves corn and why it's pretty cool. Genetics is an extremely vast field of active research, where we're constantly expanding on what we know and questioning the status quo. But like history, it's important to reflect on the major breakthroughs of our past that provided the foundations for where we are today. One of those revolutionary discoveries was made by Barbara McClintock back in the 1950s. In an experiment that she published a Nobel Prize-winning paper on, she sought to build upon the research of Rollins Adams Emerson's 1910 study on the colour of maize kernels. He had hypothesized that the spots were due to unstable mutations on the chromosomes, but McClintock had other ideas. She suspected that the colour mosaicism was due to something more complex and sought to determine what that was. But before we get into that, we need to cover some corn reproductive biology. Corn development involves a double fertilisation event, where one sperm fuses with the nucleus of the egg cell, which then produces a diploid zygote. Meanwhile, the other sperm fuses with the two polar nuclei to form a triploid endosperm, which then forms the outer protein layer known as the aleurone layer. This aleurone layer is what gives corn kernels their varying colours. This then means that the kernel colour is determined by a combination of three alleles. The first variant, the C' allele, is the dominant allele that prevents colour being expressed in the aleurone layer. The C allele, the recessive form of C', leads to colour development. When there is no C' allele, this means that the BZ alleles can then determine the colour of the kernels. The big BZ allele leads to the kernel developing purple pigmentation and is dominant over the little BZ allele, which leads to a dark brown phenotype. McClintock determined that the size and number of the differently pigmented patches of the kernels provided information on the point during development at which the genetic change took place. They also provided clear visual information, such as which cells lacked certain genes. During her study, McClintock bred females that were homozygous for the C and little bz alleles and lacked those for DS, with males that were homozygous for C', big BZ, and as she found out, a factor that she would later call DS. This then yielded heterozygotes for each of the three alleles. As each of the offspring were to inherit the C' allele, it was expected that all of the kernels were colourless, regardless of their makeup at the BZ locus. And whilst this was the case for the majority of the kernels, there were some exceptions. Some kernels had a colourless background and varying amounts of dark brown spots and streaks. McClintock, after some deliberation, concluded that some of the individual cells in those kernels must have lost their C' and BZ alleles due to a break in the chromosome, a revolutionary thought at the time. She called the site of the chromosomal breakage the DS, short for dissociation. In addition, McClintock argued that the breakage in the chromosome took place due to a factor that she called the AC, short for Activator. She found that this segment was a cut-and-paste transposon that encoded a transposase. This then interacted with the inverted terminal repeats of the AC and DS elements, and thus catalyzed the transposition and chromosome breakage that McClintock was observing. When DS was transposed along the chromosome, it had the ability to cause inactivation of neighbouring genes, and in some cases, resulted in structural instability at the sites of integration. This then caused the chromosomes to break. At the time, the AC and DS elements were called mobile genetic elements, and together formed the AC-DS system, a two-element system for controlling gene activity. Nowadays, we commonly refer to AC and DS as transposable elements. The reason why this discovery is so important now is due to its transferability to other model organisms. We now know, after almost 70 years after her publication, that transposons are the typical method of gene transfer between bacteria that confers the antibiotic resistance that we're seeing so rampant in our communities today. And in recent years, the transposition of genes from one chromosome to another have also been implicated in the process of carcinogenesis, alongside mutations that result in tumour formation. Barbara McClintock's research revolutionized the way we scientists think about the genetic patterns of inheritance, and with this, how genes are changed throughout evolution. And whilst this has made studying genetics all that more confusing and complex at times, it highlights the incredible nature of the field and just how much further we are yet to understand. For future reading, you can visit